Hello, my name is Ruslan Medzhitov. I'm a professor at Yale School of Medicine and an investigator of Howard Hughes Medical Institute. And I will discuss today our work on characterizing Toll-Like receptors and their role in control of adaptive immunity. My story started in this building. This is a library for natural sciences of Russian Academy of Sciences in Moscow when I was a graduate student there from 1990 to 93. At that time, Soviet Union just collapsed and there was a profound economic crisis, so there was no reagents to be able to do experimental work, and there were not even periodicals to be able to read. And this was the only library that still had a subscription to Nature, Science, Cell, and other journals. And so I would go there every morning and spend all day in this library on the second floor and just trying to read the literature, since I wasn't really able to do much in the lab at the time. And during one of these trips to the library, I completely randomly ran into a paper written by the late Charles Janeway that changed sort of direction of my career and life. And this was a paper that appeared in the proceedings of the Cold Spring Harbor Laboratory from 1989. And this is the paper. It was entitled Approaching the Asymptote, Evolution and Revolution in Immunology. And this is Charlie Janeway, who unfortunately passed away in 2003. But what he described in that paper was a new perspective, a new theory to think about the immune system that when I read it, I thought that this makes so much sense, and this is going to completely revolutionize our understanding of immunity. And I got completely enchanted with this idea and this logical framework that Charlie proposed there. And that was the time when I became interested in immunology. And to put this into historical context, what the idea was about is how the immune system knows when to be activated and when to respond to a challenge. And what was thought at the time is that the immune system, T cells and B cells, only react to foreign antigens, but not to self-antigens. For example, antigens that come from our own tissues. And so on the top panel, you will see here a situation where self-antigens that may be presented by antigen-presenting cells, so APCs, to T cells, but there would be no response. Even though T cells can see the antigen, they should not be responding. And at the lower part, you see the situation where there is an infection, and when APCs detect pathogens, then there should be a response. As we all know, we respond to pathogens. But it wasn't clear what makes the difference. And again, one idea was that the immune system distinguished between self-antigens and non-self antigens. So pathogens would be non-self, the immune system will react. Self-antigens are self, and the immune system would not react. But it was also known at the time that for T cells to become activated, they require two signals. And one signal is the antigen itself, which is presented by MHC molecules, major histocompatibility complex molecules. So it's a complex between MHC and antigen that is seen by T cells. And what became known at the time from the work of Ron Schwartz and Mark Jenkins at the NIH, that when T cells see only signal 1, there is no response that is generated. But when they see two signals, the second signal also coming from antigen-presenting cells, then there would be a response. And the identity of that second signal was subsequently characterized, and it's known as molecules called B71, B72, or CD80, CD86. So that was the state of knowledge, and it was many different views about what controls activation of the adaptive immune response of T cells and B cells. And there are different sort of schools of thought, and it was still very much unclear for somebody who's coming from the outside of the field what was going on and how the system worked. And that's what was changed with this article by Charlie Janeway, where he proposed that, in fact, what might be happening is that in addition to T cell receptors and immunoglobulin receptors, there is a whole different set of receptors that are shown here in blue that Charlie hypothesized would be involved in direct detection of pathogens, and that these receptors would then control expression of the signal 2. And in this manner, when antigen-presenting cells encounter self-antigens or any other antigens that are not infectious, there would be just signal 1 and there would be no response. But when they encounter pathogens, then there would be this detection by these hypothetical receptors of common microbial structures, and that would lead to the induction of signal 2, and that would result in the immune response. And that was a very profound insight, and it was a very beautiful idea that made so much sense. And Charlie further proposed that these receptors that he called pattern recognition receptors, because they detect conserved patterns found in pathogens, things like lipopolysaccharides, peptidoglycans, and so forth. He proposed that these receptors are evolutionary ancient, and they involved in immunity in invertebrates as well as invertebrates, where they control the innate arm of the immune system. But in addition, invertebrates, they acquire a second function where they control activation of the adaptive immune system. And that was really a revolutionary proposition. And the only problem was that the receptors that Charlie hypothesized should exist were not known at the time. And so after reading Charlie's paper, I started communicating with him initially through email, and eventually I was fortunate enough to be able to join his lab as a postdoc in 1994. And then my focus of my research was on trying to identify such receptors that can control expression of signal 2 to induce, to control activation of adaptive immunity. And at that time, there was really little known about how microbes can be recognized directly. There were a few proteins that were known to bind to microbial cell walls. One of them was a protein called man-non-binding lectin from the complement system. Another is CD14 molecule, which is a GPI-anchored protein on macrophages. And there are a few proteins known in invertebrates that were involved in recognition of LPS. But other than that, it was really not clear what these receptors are supposed to look like and how to identify them. And a couple of things that changed that played a role in the development of the story was that one was in 1994, the year when I joined Charlie Lab. David Baltimore came to Yale to give a talk at our department. And at that time, his lab just started characterizing the first knockouts for NF-kappa B, which is a critical transcription factor involved in inflammation. And there he talked about the phenotype of first NF-kappa B knockouts, which had both defects in innate and an adaptive immune response. And I remember after David's talk, Charlie and I were talking in the hallway, and we both thought that whatever it is that we are looking for, whatever these receptors are, they probably work by activating NF-kappa B. And then we thought, okay, so one criteria we should use is that they are likely to be NF-kappa B activators. And at that time, the only two receptor families known to activate NF-kappa B were TNF family and IL-1 receptor family. And IL-1 receptor is the one that we got particularly interested in because of this remarkable conservation in IL-1 receptor signaling portion with a receptor that at the time was already known to exist in Drosophila. And in Drosophila, that receptor is called Toll. That receptor was identified and cloned in Catherine Anderson's lab by Carl Hashimoto. At the time, they were at Berkeley. And the signaling pathway was elucidated by several investigators, including Steve Wasserman and Mike Levine, Catherine Anderson, and others. And it was known that this pathway operates in fly development. But what was interesting for us is that it worked through NF-kappa B in flies, and the related receptor, IL-1 receptor, worked through NF-kappa B in mammals. And what they shared is the cytoplasmic portion shown here in blue, whereas their ectodomains involved in ligand recognition were unrelated. And because of this conservation with flies, and because IL-1 receptor is involved in inflammation, we thought that perhaps the receptor that we are looking for would have this C-terminal signaling domain, and the ectodomain perhaps would be something that can recognize microbes. And the only structures that were known at the time to recognize microbial components were C-type lectins. So I thought that maybe there is another version of this receptor that has cytoplasmic signaling domain from Toll and IL-1 receptor, and the ectodomain would be from C-type lectin. And that was my strategy to try to identify it. And what I took advantage of is that after many failed attempts to do it through various types of approaches for degenerate screening or by hybridization or through PCR, took advantage of the fact that at the time new types of genomic sequences started to become available, these so-called express sequence tags, that were just short sequences that were generated randomly in multiple tissues. And using bioinformatic approaches and using conserved consensus sequences for the cytoplasmic domain for Toll and IL-1 receptor, as well as for C-type lectins, I started searching these databases and found several clones that corresponded either to C-type lectins or to this cytoplasmic domain. And then I pursued both of them, and the one that corresponded to C-type lectin domain, I found that it was most similar to Drosophila toll. And this was in starting in January, February in 1996, and the gel shown on the left here was, I think, one of the first clones that I got, which would later would turn out to be a human homologue of Toll. And the gel on the right and in this bottom band, I still remember that band because this was one of those happy moments in the lab when I got this portion of the receptor which was particularly difficult to clone. And then by doing standard library screening through hybridization, we pulled out a full length of the clone encoding Toll receptor, followed by 5' raise technique, which was at the time popular in gene cloning, and eventually got the receptor. And by about May of '96, and then another event that happened that in the summer of '96, we had a meeting in Charlie's summer house in Annisquam near Boston, where several other investigators participated that shared Human Frontier Grant, including Alan Zakowicz, Fotis Skafadis, Jules Hoffman, and Charlie. And Jules Hoffman then presented work of Bruno Lemaitre from his lab about genetic work on Drosophila toll pathway, where they found it was involved in immunity. So that was gave us further confirmation for our expectation that this receptor might be involved in immunity. And so then I characterized this receptor in terms of its ability to control NF-kappa B signaling, shown here on the right. And more importantly for us at the time, for its ability to induce inflammatory cytokines, and the most important for us, the holy grail for us, whether this receptor can induce the second signal or co-stimulatory molecule, shown as B7. And that was probably one of those lucky moments in the lab when I saw that Toll receptor, which I rendered constitutively active because the ligand was not yet known at the time, and transfected in this monocyte cell line. And when I saw that it can induce expression of this gene, that was the moment when we thought, okay, this whole hypothesis now we can connect from beginning to the end. And I even called Charlie. This was late in the evening. I even called Charlie and told him that that human Toll can induce B7 expression. We probably would be the only two people in the world who would be excited about that. But nevertheless, others also appreciated it, and so we published this paper in '97, which is extremely simple paper showing just cloning and showing this couple of functional assays. And then subsequently, in the same year, Ben Lune, who was the editor of Cell at the time, asked Charlie to write a mini review about innate immunity. And so in that mini review, we discussed questions about evolution of innate and adaptive immunity, and put this schematic together to show the parallels and differences between Drosophila Toll and mammalian Toll pathways, where we suggested that recognition of pathogen-associated molecular patterns, things like LPS and teichoic acids and so on, is detected by Tolls either directly or through some intermediary, as is the case in Drosophila, and that leads to induction of NF-kappa B and various innate immune response genes and genes controlling adaptive immunity. And subsequent work indeed demonstrated that the Toll that I worked on is part of a bigger family. There are about a dozen of different Toll receptors in mammalian species, and it's now known as TLR4, Toll-Like Receptor 4, and it's a receptor for LPS. And the receptor part that detects LPS, actually a protein called MD2 that complexes with Toll. And subsequent work by many investigators elucidated specificities of different Toll receptors, and Shizuo Akira from Osaka University in particular played a major role in this set of investigations. And what we know now is that our main interest was not so much in microbial specificity of receptors, but in this idea that microbial receptors can control activation of adaptive immunity. And this is what schematically illustrated here. So we have a dendritic cell, which is a cell type that normally activates T cells. And when dendritic cell encounters pathogen, what's shown on the left side here, there is an endocytic receptor that will take up pathogen, take it into lysosomes, and then proteins will be cut into peptides and loaded into MHC molecules and presented to T cell receptors. But that information by itself is not sufficient for T cells to know whether they should become activated or not, because this peptide can come from pathogen, or it can come from innocuous food antigens, or it could be even self-antigen. So T cells have no way of knowing what's the origin of the antigen is. And the reason for that is because T cell receptors are generated at random. And each T cell has one single specificity, but they are random, so it doesn't know what it's specific for. So therefore, there is a need for another signal, so-called signal 2, which will provide information about the origin of the antigen that T cell is specific for. And that is provided by this detection of microbial structures by Toll receptors, and now we know several other families of pattern recognition receptors that detect those structures and induce expression of costimulatory molecules. And now T cell that has specificity for peptide derived from pathogen will also have a confirmation signal from costimulatory molecule CD80, CD86, also known as B71, B72, and then T cell will become activated. And in addition, Toll receptors and other pattern recognition receptors will induce production of cytokines such as IL-12 that will tell T cells what kind of effector response to generate. And that is now, of course, a well-accepted view how immune response is activated. and it's a very satisfying to see this confirmation of the proposal by Charlie Janeway, which was at the time largely ignored, and that now it's a textbook knowledge of the immune system. That's how activation of the adaptive immune system is controlled. And Toll receptor was one of the first receptors to demonstrate it to be involved in this process. So that's the story of this discovery, and thank you for listening.